Coming to our next topic, lipid metabolism. So in this we are going to discuss about the main pathway in lipid metabolism, that is oxidation of fatty acids. So lipids, they serve as the important sources of energy. So lipids, neutral lipids, they are nothing but triacylglycerols. They have esterified fatty acids. Those fatty acids, they release energy by a process called as oxidation. So if the oxidation occurs on the beta carbon of a fatty acid, we call it as beta oxidation. If it is alpha carbon, we call it as alpha oxidation. So depending on the carbon which is oxidized, so we have different types of oxidation. So major type of oxidation of fatty acids is beta oxidation. So here the oxidation of the beta carbon occurs with the removal of two carbons from the beta carbon is called as beta oxidation. So here oxidation of the beta carbon is occurring with the removal of two carbon atoms in the form of acetyl-CoA occurs in beta oxidation. So this is what we can define beta oxidation as. Coming to its site where this beta oxidation occurs. So it occurs in both cytosol and mitochondria. So the major part of the beta oxidation of fatty acids occurs in the mitochondria, whereas the most important activation occurs in the cytosol. So there is only one reaction that occurs in the cytosol, and all the remaining reactions, they occur in the mitochondria. Right. Coming to stages. So when you see beta oxidation, the process of whole beta oxidation, we can divide it into three parts. Right. So one is activation of fatty acids. So in order to derive the extract energy from the fatty acids, first we have to activate that fatty acid. So first step in the beta oxidation of fatty acids is activation of fatty acids. So second one is, so after its activation we have to transport that fatty acid into the mitochondria. And the third stage is the beta oxidation proper. So the remaining part of the beta oxidation occurs in the mitochondria. So these are the three stages of beta oxidation, that is activation of fatty acids, transport of fatty acids into mitochondria, and the beta oxidation proper. And so first we'll see how this fatty acid is activated. So the activation of fatty acid is brought by an enzyme called as acyl-CoA synthetase. So here we are activating the fatty acid by adding a coenzyme A, right? So this is how the fatty acid is activated, and for this we require the energy and supplied by the adenosine triphosphate, ATP, right? So this acyl-CoA synthetase, also called as thiokinase, is involved in the activation of the fatty acids. So this step occurs in the cytosol of the cell, right? So cytosol, the only step that occurs in case of beta oxidation in cytosol is activation of fatty acids. So fatty acid is activated into corresponding acyl-CoA. So if it is palmitic acid, the activated form of palmitic acid is called as palmitoyl-CoA. So likewise, depending on the fatty acid, we have its active form with a coenzyme added to that fatty acid. Right? So palmitic acid and its active form is palmitoyl-CoA. So in general, if it is fatty acid, we call it as acyl-CoA. Right? So this is how activation of fatty acids occurs. So once it is activated to acyl-CoA, now that has to be transported into the mitochondria, because acyl-CoA cannot cross the mitochondrial membrane. We require a special type of transporter called as Carnitine Transporter. So carnitine is made up of lysine and methionine. So this carnitine is involved in the transport of activated fatty acids from cytosol into the mitochondria. That is the use of carnitine in the beta oxidation. So it transports the activated fatty acid which cannot cross the mitochondrial membrane into the mitochondria. So without carnitine, fatty acid oxidation cannot occur because it cannot cross the mitochondrial membrane. So carnitine transports the acyl-CoA from cytosol into the mitochondria. So now activated fatty acid, that is acyl-CoA, has entered the mitochondria. Now what happens in the mitochondria? We'll see So in the mitochondria, now acyl-CoA entered into the mitochondria. In mitochondria, it undergoes oxidation catalyzed by the enzyme called as acyl-CoA dehydrogenase. Right. So that acyl-CoA dehydrogenase is catalyzes the first oxidation step in the beta oxidation. So that here the coenzyme required is FAD, and that is reduced to FADH2. Right. So by the action of this enzyme acyl-CoA dehydrogenase, acyl-CoA is converted into beta hydroxy. It is converted to trans-enyl-CoA. So delta 2 trans-enyl-CoA is formed by the action of enzyme called as acyl-CoA dehydrogenase. And in the next reaction, this delta 2 trans-enyl-CoA by the action of enzyme called as enyl hydratase. So hydration, water molecule added in this reaction by the action of enzyme called as hydratase, so that enyl hydratase is converted into beta hydroxy acyl CoA or 3-hydroxy acyl CoA. So whatever it may be. So this hydroxy acyl CoA in the next step catalyzed by the enzyme called as hydroxy acyl CoA dehydrogenase. So this is the second step in beta oxidation where oxidation occurs. So that is beta hydroxy acyl CoA is converted to keto acyl CoA by the action of enzyme called as acyl CoA with hydroxy acyl CoA dehydrogenase. And here the coenzyme required is NAD plus, and that is reduced to NADH. Now what happens to that keto acyl CoA? That keto acyl CoA in the next reaction catalyzed by the enzyme called as thiolase, and that converts by a process called as thiolysis. It breaks the keto acyl CoA into two parts. So one is acyl CoA minus 2 carbons, and those 2 carbons are removed in the form of acetyl CoA. So in a single turn of beta oxidation, 2 carbons are removed in the form of acetyl CoA. So sequentially we will remove 2 carbons in the form of acetyl CoA, and that occurs in the beta oxidation. So oxidation of the beta carbon and splitting of 2 carbons in the form of acetyl CoA is what happens in case of beta oxidation, and whatever the acetyl CoA that is released, now that enters into the TCA cycle, citric acid cycle, where it is further oxidized. So this is the beta oxidation proper. Right? So here 2 oxidation reactions occur. We require 2 dehydrogenases, and the first one is acyl CoA dehydrogenase. There the enzyme is coenzyme is FAD, and the other one is beta hydroxy acyl CoA dehydrogenase. Here the coenzyme is NAD. So actually the FAD is reduced to FADH2 and NAD plus is reduced to NADH. So these reduced coenzymes, they enter into the electron transport chain, where they donate these electrons to the components of electron transport chain, so that the flow of electrons to these complexes occurs, and that results in the synthesis of ATP. Right? So this is what happens to the FADH2 and NADH generated in the oxidation of fatty acids. Now coming to energetics, so as I told you, so in this slide you can see FADH2 and NADH are generated in the oxidation proper. So that FADH2 and NADH, they enter into the electron transport chain, and when NADH enters into the electron transport chain, it generates or produces the ATPs equivalent to 2.5 according to our new calculations. You can consider 3 ATPs. Right? So when you consider one NADH can generate 3 ATPs and FADH2 can generate 2 ATPs. And when you consider palmitic acid oxidation, there 16 carbon fatty acid. So when it enters into the beta oxidation, it has total 16 carbons, and total 8 acetyl CoAs are produced and the number of cycles, beta oxidation cycles, they occur is 7 cycles. So 7 turns of beta oxidation occurs, and total 8 acetyl CoAs are produced, and 7 cycles of beta oxidation will produce 7 FADH2 and 7 NADH. So 7 FADH into 3 and 7 NADH into 3. So there you can calculate the number of ATPs from NADH and FADH2 generated in the beta oxidation. And coming to another part, so at the end of total cycles, 7 cycles, will get total 8 acetyl-CoA. Those 8 acetyl-CoA, now they enter into the electron, they enter into the TCA cycle. So where it undergoes the oxidation and it produces 12 ATPs. So when a single acetyl-CoA enters into the TCA cycle, produces 12 ATPs, and in case of beta oxidation of palmitic acid, total 8 acetyl-CoAs are produced. So 8 into 12 ATPs will be generated here. Right. So when you calculate, so this is about the number of ATPs produced, and actually in case of activation 1 ATP is utilized and that is converted into AMP. So we'll calculate that, we'll consider that as utilization of 2 ATPs, and the total number of ATPs produced here, it is 131 ATPs, and in case of activation we use 2 ATPs. So ATPs utilized are 2, and the net gain of ATPs is 129 ATPs. So this is about the energetics of beta oxidation, and that is the significance of beta oxidation. It is mainly concerned with the generation of energy, and that's about the beta oxidation. Coming to next topic, metabolism of ketone bodies. So what are ketone bodies? So acetone, acetoacetate and beta hydroxybutyrate are called as ketone bodies. So they have a ketone group, because of that they are called as ketone bodies. So though beta hydroxybutyrate is not a, it has no keto group. So though it has no keto group, we call it as ketone bodies because so it can serve as a precursor for the synthesis of ketone body acetoacetate. So these three, acetone, acetoacetate and beta hydroxybutyrate, these three are called as ketone bodies or acetone bodies, right? And ketogenesis and ketolysis. When you see, metabolism of ketone bodies involves two processes, ketogenesis and ketolysis. So what is ketogenesis? Synthesis of ketone bodies, formation of ketone bodies is called as ketogenesis. And when you see, so this process occurs in the liver mitochondria, and here the precursors for the ketone body synthesis are acetyl-CoA. So we require 2 molecules of acetyl-CoA and 2 molecules of acetyl-CoA, they condense to form acetoacetyl-CoA, and that is catalyzed by the enzyme called as thiolase. Now that acetoacetyl-CoA again reacts with one more molecule of acetyl-CoA, and this reaction is catalyzed by an enzyme called as HMG-CoA synthase, and that results in the synthesis of a product called as, that is the formation of a compound called as beta hydroxy beta methyl glutaryl CoA. In short, we call it as HMG-CoA. Now what is the fate of that HMG-CoA? Now that HMG-CoA in the next reaction undergoes lysis catalyzed by an enzyme called as HMG-CoA lyase. So one acetyl-CoA is removed from the beta hydroxy beta methyl glutaryl CoA so that it is converted into acetoacetate. Now that acetoacetate is called as primary ketone body, right? And that acetoacetate undergoes spontaneous decarboxylation to form acetone, another ketone body, and that undergoes one more reaction, and that is catalyzed by the enzyme called as beta hydroxybutyrate dehydrogenase, so that acetoacetate can be reversibly converted into beta hydroxybutyrate, right? So these three, acetoacetate, acetone and beta hydroxybutyrate, these three are called as ketone bodies or acetone bodies, right? So we require the simple precursors acetyl-CoA can be used for the synthesis of ketone bodies. Coming to ketolysis. So we discussed that ketone bodies metabolism involves ketogenesis and ketolysis. So what happens in ketolysis? In ketolysis, we are utilizing the ketone bodies, right? So utilization of ketone bodies is nothing but ketolysis. So how that ketolysis occurs? That ketolysis occurs in all extrahepatic tissues. Ketogenesis occurs in the liver, but ketolysis, utilization of ketone bodies, occurs in the all extrahepatic tissues because liver lacks the enzyme which is involved in the utilization of ketone bodies. That is, so there is no enzyme thiophorase in the hepatocytes in the liver, so that it cannot utilize the ketone bodies. Though it synthesizes ketone bodies, liver cannot utilize ketone bodies because of deficiency of enzyme called as thiophorase. So it lacks that particular enzyme, so that's why it cannot utilize the ketone bodies. So otherwise all the extrahepatic tissues, they can utilize the ketone bodies. So mostly heart muscle, and that is the main tissue which utilizes the ketone bodies. And when you see nucleation of ketone bodies, so as I told you, there is one enzyme which can reversibly convert esteroid into beta hydroxybutyrate into esteroid. That is beta hydroxybutyrate dehydrogenase. So that beta hydroxybutyrate dehydrogenase is converted into esteroid, and esteroid can be converted into a product called esteoacetyl-CoA. So esteoacetate is converted into esteoacetyl-CoA. In this reaction, this reaction is catalyzed by an enzyme called as thiophorase and requires succinyl-CoA. Right? So that donates the coenzyme A to the esteoacetate, so that is converted into acetoacetyl-CoA. So once it is converted into acetoacetyl-CoA, and that acetoacetyl-CoA by the action of enzyme called as thiolase, so thiolytic cleavage occurs here, thiolytic lysis occurs, and in this reaction two molecules of acetyl-CoA are produced, and this thiolase requires coenzyme A. Right? So this is how esteoacetate beta hydroxybutyrate, they can be converted into acetyl-CoA. And once it is converted into acetyl-CoA, that acetyl-CoA enters into the PCA cycle, and PCA cycle it is subjected to oxidation. By oxidation, it produces generates the reducing coenzymes, and those reducing coenzymes, they enter into the electron transport chain, so thereby they can generate the ATPs by a process called as oxidative phosphorylation. So this is how utilization of ketone bodies occurs. So the main enzyme required in case of utilization of ketone bodies is thiophorase. So it is absent in the livers. All the extrahepatic tissues, they have this thiophorase. Now coming to the clinical condition associated with ketone bodies metabolism is ketosis. So ketosis is nothing but ketonemia plus ketonuria plus acetone in breath. So ketonemia is, so whenever there is increased concentration of ketone bodies in the blood, we call it as ketonemia, and the normal concentration is 1 milligram per deciliter. Right? So whenever it is more than 2 milligrams per deciliter, generally we call it as ketonemia, and so whenever it is more than 1 milligram per deciliter, more than 2 milligrams per deciliter, so that will be excreted through urine. That condition we call it as ketonuria. So excess ketone bodies in blood, we call it as ketonemia, and whenever it is in excess, that is excreted through urine, we call it as ketonuria. And acetone, which is formed from the esteoacetate by spontaneous decarboxylation, is a volatile ketone body, and that is expired in, so that is excreted through the lungs. So these three conditions, ketonemia, ketonuria and acetone in breath, these three together we call it as ketosis. Right? So this is about the ketosis. So what causes ketosis? Generally in case of severe untreated diabetes mellitus, so ketosis is seen, and apart from that in case of starvation, prolonged starvation also, ketosis is seen. Coming to next, cholesterol metabolism. So this cholesterol is synthesized in our body in all the nucleated cells, and the major site where it is synthesized is the liver. Right? And when you see the enzymes of concern with the cholesterol synthesis, they are located in the cytosol and endoplasmic reticulum. And precursor, here the simple precursor is acetyl-CoA. So all the carbons, so cholesterol has 27 carbons, all the 27 carbons of cholesterol, they are derived from the 2-carbon compound, that is acetyl-CoA. Now coming to steps of cholesterol synthesis. So there are 5 steps in the cholesterol synthesis. So that is first, synthesis of a 6-carbon compound called as malonic acid, and next formation of isoprenoid units. So those isoprenoid units, they can be converted into a 30-carbon compound, linear squalene. So linear squalene undergoes cyclization to form lanosterol, and that lanosterol is further processed to form cholesterol. So these are the 5 steps in the cholesterol synthesis. So here, so as we discussed, the precursor is acetyl-CoA. So two acetyl-CoAs, they react to form esteoacetyl-CoA by the action of enzyme called as thiolase, and S2-acetyl-CoA in the next reaction catalyzed by an enzyme called as HMG-CoA synthase is converted to HMG-CoA, beta-hydroxy beta-methyl glutaryl-CoA, and that beta-hydroxy beta-methyl glutaryl-CoA in the next reaction catalyzed by the enzyme called as HMG-CoA reductase. So this HMG-CoA reductase is the key enzyme of cholesterol synthesis. So what happens to that HMG-CoA? So HMG-CoA reductase acts on HMG-CoA and converts that into a compound called as mevalonic acid or mevalonate. So that mevalonic acid is next processed, converted into isoprenoid unit, that is active form of isoprenoids, that is isopentenyl pyrophosphate by 3 kinases, by reaction of 3 kinases. Alright, so mevalonate undergoes 3 phosphorylations catalyzed by enzyme called as kinase. So mevalonate kinase converts that into mevalonate 5-phosphate. Next, one more by the action of one more kinase, it is converted into mevalonate 5-pyrophosphate, and one more kinase acts on that mevalonate 5-pyrophosphate, converts that into mevalonate 3-phospho-5-pyrophosphate. So by the action of 3 kinases, mevalonate is converted into mevalonate 3-phospho-5-pyrophosphate. So once it is converted into mevalonate 3-phospho-5-pyrophosphate, it undergoes decarboxylation and is converted into isopentenyl pyrophosphate. So that isopentenyl pyrophosphate now has 5 carbons. So actually mevalonate has 6 carbons. So whenever it undergoes decarboxylation, it is now converted into a 5-carbon compound called as isopentenyl pyrophosphate. That isopentenyl pyrophosphate now undergoes isomerization to form dimethyl allyl pyrophosphate. So these two are the 5-carbon compounds, these two are isomers. Next, so in the next reaction, isopentenyl pyrophosphate reacts with dimethyl allyl pyrophosphate and is converted into a 10-carbon compound called as geranyl pyrophosphate. Next, so once geranyl pyrophosphate is formed, so that geranyl pyrophosphate in the next reaction reacts with one more molecule of isopentenyl pyrophosphate and is converted into a 15-carbon compound called as farnesyl pyrophosphate. So that farnesyl pyrophosphate, so 2 molecules of farnesyl pyrophosphate, they react to form a 30-carbon compound called as squalene. Squalene, by the action of enzyme called as squalene monooxygenase, is converted into an epoxide form, and that epoxide form is converted into lanosterol by the action of enzyme called as squalene epoxidase. So that epoxidase converts epoxide form of squalene into lanosterol. So that lanosterol is a 30-carbon compound, and that in the next sequence of reactions, about 19 reactions they occur, and during these reactions 3 carbons are removed, and finally a 27-carbon compound cholesterol is synthesized in the reactions. So this is how a simple 2-carbon compound S2-CoA is used in the synthesis of a 27-carbon compound, that is cholesterol. Right? So this is about the synthesis of cholesterol. Now coming to regulation of cholesterol, how this cholesterol synthesis is regulated, so that we can discuss under 3 headings: feedback, inhibition, proteolytic degradation, and the other one is hormonal regulation. So by these 3 ways, the cholesterol synthesis can be regulated. Coming to feedback inhibition, so whenever the concentration of cholesterol is more, that product, the end product of cholesterol synthesis, cholesterol, inhibits the key enzyme of cholesterol synthesis, that is HMG-CoA reductase. So in this way, cholesterol synthesis is regulated. And coming to another type of regulation, proteolytic regulation. So whenever the concentration of cholesterol is more, then it results in the synthesis of a molecule called as INSIC. So that INSIC mediates the proteolytic degradation of HMG-CoA reductase. So that INSIC adds the ubiquinone to this HMG-CoA reductase, and that leads to proteolytic degradation of HMG-CoA reductase, and by this way, so cholesterol synthesis can be regulated. Coming to hormonal regulation, so hormones like glucagon, they add a phosphate group to the key enzyme, that is HMG-CoA reductase. So whenever a phosphate group is added to HMG-CoA reductase, the activity of HMG-CoA will be decreased, right? So phosphorylation decreases the activity of HMG-CoA, and that is brought by glucagon. So whenever it is undergoing phosphorylation, HMG-CoA activity decrease, it is inactivated, and the cholesterol synthesis will be inhibited, right? And other hormone, insulin, favors the dephosphorylation. So whenever the phosphate group is removed, the activity of HMG-CoA is retained, the activity is increased, and that results in the increased synthesis of cholesterol. So this is the effect of hormones on the regulation of cholesterol synthesis. So glucagon decreases the cholesterol synthesis, insulin increases the cholesterol synthesis by phosphorylation, and insulin by dephosphorylation. So this is about the regulation of cholesterol synthesis, and there are some important compounds derived from the cholesterol like vitamin D is derived from the cholesterol, and steroid hormones, bile acids, estrogen, progesterone. So all these compounds, mineralocorticoids, glucocorticoids, so these are the important compounds derived from the cholesterol, right? So what are those important compounds? Those are vitamin D and bile acids, mineralocorticoids, glucocorticoids, progesterone, estrogens are the important compounds derived from the cholesterol. Coming to clinical significance of cholesterol. So the normal concentration of cholesterol in the blood is 150 to 200 milligrams per deciliter, and the important fractions of cholesterol, so those are HDL cholesterol and LDL cholesterol. So this type of cholesterol, HDL cholesterol, is called as good cholesterol because it prevents the accumulation of cholesterol in the arteries. So when you see the concentration of HDL cholesterol in the blood is 30 to 60 milligrams per deciliter in case of males, and it is slightly more in case of females, and its concentration is 35 to 75 milligrams per deciliter. So in case of LDL, so LDL is called as bad cholesterol because it is an atherogenic agent. So it is responsible for the atherosclerosis. So whenever the concentration of LDL is more, it leads to the risk of atherosclerosis increases. And LDL concentration of LDL is it is 60 to 150 milligrams per deciliter, and the ideal concentration is it should be less than 150 milligrams per deciliter, and for the HDL level it should be more than 60 milligrams per deciliter. It should be above 30 milligrams per deciliter, and when you see this LDL cholesterol, it should be below 150 milligrams per deciliter. So that is the normal ranges of cholesterol, total cholesterol, HDL cholesterol and LDL cholesterol. Coming to hypercholesterolemia, whenever the concentration of cholesterol is more than 200 milligrams per deciliter, so generally we call that condition as hypercholesterolemia. And so in case of that hypercholesterolemia, see in case of diabetes mellitus, nephrotic syndrome, obstructive jaundice, hypothyroidism. So these are the conditions in which it leads to hypercholesterolemia. So in case of diabetes mellitus, so there is deficiency of insulin. Whenever there is deficiency of insulin, so body depends upon other sources like fatty acids. There is increased lipolysis, and due to increased lipolysis, more fatty acids are oxidized, and that leads to synthesis of ester, that leads to formation of excess amount of acetyl CoA, and that acetyl CoA is used in the synthesis of cholesterol, right? When you see in case of obstructive jaundice, there is obstruction in the excretion of bile, so that leads to increased concentration of cholesterol in the blood. So that is one more condition that leads to hypercholesterolemia. And coming to hypothyroidism and nephrotic syndrome, those two are also in these two conditions also there is increased concentration of cholesterol in the blood. So this is about the cholesterol metabolism. Coming to lipoproteins. So what are lipoproteins? They are conjugated proteins. So they have two parts, protein part plus non-protein part, and the non-protein part of lipoproteins is lipids. Lipids plus proteins, that is a composition of lipoproteins. And the protein part of lipoprotein is called as apoprotein. There are different types of apoproteins and in different types of lipoproteins, right? And when you see the functions of lipoproteins, the main functions of lipoproteins is the transportation of lipids. So they transport various lipids in the blood. That is the function of lipoprotein. Coming to types of lipoproteins, we have different types of lipoproteins, 5 types. So those are chylomicrons, VLDL, IDL, LDL and HDL. So chylomicrons, VLDL is very low density lipoprotein, IDL is intermediate density lipoprotein, LDL is low density lipoprotein, HDL is high density lipoprotein. So these are the 5 types of lipoproteins. And coming to their separation, these lipoproteins can be separated by 2 techniques. One is ultracentrifugation and the other one is electrophoresis. Right? So the first ultracentrifugation is based on the density of the lipoproteins, and the least dense is chylomicrons. The content of fat is more in case of chylomicrons, so that's why it floats. So the first fraction in case of ultracentrifugation is chylomicrons, and then comes the VLDL, IDL, LDL, HDL, and the last fraction is free fatty acids. So this is how we can separate the lipoproteins by ultracentrifugation, and that is based on the density. Now coming to the second way of separation of plasma lipoproteins, that is by electrophoresis. Here it depends on the charge of the lipoproteins. So those which have more amount of proteins, generally they tend to move faster in the electrophoresis than those which have less protein content. Right? So when you see the fastest moving fraction in case of electrophoresis is free fatty acid soluble complex, then comes the HDL cholesterol, VLDL, IDL, LDL, and the last and the least mobile fraction is the chylomicrons, because it has high fat content and less protein content. So that's why this is the least moving fraction. So it remains at the site of application of the sample, and the fastest moving fraction is the albumin free fatty acid albumin complex, and HDL is the next fraction. So this is how lipoproteins can be separated by 2 techniques, ultracentrifugation and electrophoresis. Coming to functions of each lipoprotein, so when you see chylomicrons, so they are synthesized in the intestine. The function of chylomicrons is, so this chylomicrons is involved in the transport of exogenous TAC, means whatever the lipids we take in the diet, they are transported from the intestine to the adipose tissue and to the remaining tissues like skeletal muscle for utilization. So the dietary TAC, exogenous TAC, is transported from the intestine to the peripheral tissues by chylomicrons. So that is the function of chylomicrons. And coming to VLDL, so this VLDL is synthesized in the liver, and the function of VLDL is, it is also involved in the transport of triacylglycerols, but it transports the triacylglycerols from the liver to the other tissues, peripheral tissues. Okay? So that is the function of VLDL. So it also transports triacylglycerols, but it transports endogenous triacylglycerols. So chylomicrons transports exogenous triacylglycerols, whereas VLDL transports the endogenous triacylglycerols. Whatever synthesized in our body, in the liver, they are transported to the tissues, peripheral tissues by the VLDL. Coming to LDL, so it is synthesized from the VLDL. Its function is, it transports the cholesterol from the liver to the extrahepatic tissues. So that is the function of LDL. Right? Coming to the HDL cholesterol, high density lipoprotein, HDL, high density lipoprotein. The function of high density lipoprotein HDL is, it transports the cholesterol from the extrahepatic tissues, from the peripheral tissues to the liver. So this type of transport we call it as reverse cholesterol transport. So this is involved in transport of cholesterol from the extrahepatic tissues to the liver. We call it as reverse cholesterol transport, and that is brought by the HDL. So that is the function of HDL lipoprotein. Coming to disorders of lipoprotein metabolism. Right? So whenever there is some defect in the lipoprotein metabolism, it leads to increased concentration of lipoproteins. If there is increased lipoproteins in the blood, we call it as hyperlipocrotinemia. So if it is decreased concentration of lipoproteins in the blood, we call it as hypolipoproteinemia. Coming to the most important, hyperlipoproteinemia, we have type 1 hyperlipoproteinemia, also called as familial hypercholesterolemia, and this is because of deficiency of enzyme called as lipoprotein lipase (LPL), otherwise because of deficiency of ApoC2. So that activator of lipoprotein lipase leads to this type of hyperlipoproteinemia called as type 1 hyperlipoproteinemia or familial hypercholesterolemia. So whenever there is defect in this particular lipoprotein lipase or the activator of that particular lipoprotein lipase ApoC2, it leads to increased triacylglycerols in the blood. So that is called as hypertriglyceridemia. So there is increased triacylglycerols in the blood. That is the manifestation of type 1 hyperlipoproteinemia. Coming to type 2 hyperlipoproteinemia, that is called as type 2 familial hypercholesterolemia. In this condition, so here the defect is defect in the LDL receptors. So whenever there is lack of LDL receptors is there, so that LDL receptor deficiency leads to increased concentration of LDL in the blood, and that is the important feature of type 2 familial hypercholesterolemia. Right? So concentration of LDL cholesterol increases in the blood. Right? Coming to atherosclerosis, right? So what is atherosclerosis? So it is a condition in which there is hardening of the hardening and thickening of the arterial walls generally seen because of deposition of cholesterol. So atherosclerosis is hardening of the arteries. So that hardening and thickening of the arteries is because of the deposition of cholesterol. So that process we call it as atherosclerosis. So here you can see the normal artery, and whenever there is atherosclerosis, deposition of fat, deposition of cholesterol, and that leads to blocking that narrows the arteries and that obstruct the flow of blood. So that is important feature of atherosclerosis, that obstructs the flow of blood through the arteries. And here you can see so the mechanism of atherosclerosis, how it occurs. So whenever the oxidized LDL is there, that oxidized LDL so enters into the intima, and that oxidized LDL attracts the macrophages. So whenever oxidized LDL is there, monocytes are attracted to that site, and those monocytes are transformed into macrophages, and those macrophages, they engulf these oxidized LDL and they are converted into foam cells. So those foam cells, they release some growth factors, and because of those growth factors, it leads to formation of a fibrous tissue like thing, and that leads to narrowing of the arteries, and finally that condition we call it as atherosclerosis. Right? So oxidized LDL is mainly responsible for this atherosclerosis. Coming to progression of atherosis, how this atherosclerosis occurs. So what are the risk factors? So risk factors of atherosclerosis are hyperlipidemia. Whenever the concentration of lipids are more, it is the main factor which is responsible for the atherosclerosis. And hypertension, diabetes mellitus, smoking, and sedentary lifestyle and obesity are these are the important reasons which results in the atherosclerosis. Coming to another important aspect of lipid metabolism, fatty liver. So whenever there is increased accumulation of fat in the liver, so that condition we call it as fatty liver. So what is fatty liver? Increased accumulation of fat in the liver is called as fatty liver. And coming to causes, what causes fatty liver? So there are two causes, that is whenever there is excess deposition of fat in the liver, so that leads to fatty liver, and other reason is reduced removal of fat from the liver. So these two conditions will lead to fatty liver. Now coming to first one, excess fat deposition in the liver. Right? So why this excess deposition of fat occurs in the liver? Because whenever there is increased mobilization of fat from the adipose tissue, that leads to increased accumulation of fat in the liver. Right? So that is one reason, and the other reason is more synthesis of fatty acids from the glucose. Whenever you take more amount of glucose in the diet, so that glucose can be used in the synthesis of fat, so that excess calories whatever you are taking, those are converted into fat, and that is one more reason, so because of which it leads to excess fatty acid synthesis, and that gets deposited in the liver, right? So this is the reason, first reason for the fatty liver, that is excess deposition of fat in the liver. So that is mainly because of excess mobilization of fat from the adipose tissue, and there is excess synthesis of fat from the glucose. Whatever you are taking in excess, that can be converted into fat. And coming to next reason, second cause, it is reduced removal of fat from the liver. So whenever you liver fails to remove the fat from the liver, that leads to increased deposition of the fat in the liver, and that results in fatty liver. So what are the main causes? So that leads to reduced removal of fat from the liver. So those are, so whenever there is injury to the liver, toxins like carbon tetrachloride, salicylates, so all those toxic compounds, whenever they damage the hepatocytes liver, it leads to decreased synthesis of apoproteins, which are required for the excretion of fat in the form of VLDL. So triacylglycerols, they are transported from the liver to the peripheral tissues by VLDL. For the synthesis of VLDL, we require the apoproteins. So whenever hepatocytes are damaged, the synthetic capacity of the liver decreases, so less amount of apoproteins are synthesized, and that leads to decreased VLDL synthesis, and fat cannot be transported out of the liver, and that leads to accumulation of fat in the liver. So that is the reason. So whenever toxic injury of liver occurs, it leads to excess deposition of fat in the liver. And the second reason is decreased oxidation of fat by the hepatocytes. So that also leads to fatty liver, right? So whenever these two conditions occur, it leads to increased accumulation of fat in the liver. So when you see, so there are some important factors which prevent the accumulation of fat in the liver, or which increase the mobilization of fat from the liver, which prevent the accumulation of fat in the liver. So those factors, we call them as lipotrophic factors. So those factors which prevent the accumulation of fat in the liver. So that is the main function of those factors. We call all those factors together as lipotrophic factors, like choline, betaine, methionine, right? Omega-3 fatty acids. So all these are called as lipotrophic factors because they prevent the accumulation of fat in the liver. And the mechanism by which they prevent the accumulation of fat in the liver, we call it as lipotropism. So this is the factors. This is about the factors which prevent the accumulation of fat in the liver. Right. So till now we have seen how the fatty acids are oxidized in our body, how cholesterol is synthesized, what are lipoproteins, what is their functions, and what is the atherosclerosis, and what is fatty liver, what causes fatty liver, and what are the factors which prevent the accumulation of fat in the liver. Those are called as lipotrophic factors. Thank you.